Hello, students. Here is Genetic Engineering Class, Chapter 3, presented at Nanjing Forestry University. I am Dr. Ali Mowahedi. Here we go. In this chapter, we want to talk about working with nucleic acids. Before examining some of the specific techniques used in gene manipulation, it is useful to consider the basic methods required for handling, quantifying, and analyzing nucleic acid molecules. It is often difficult to make the link between the theoretical and practical aspects of a subject, and an appreciation of the methods used in routine work with nucleic acids may be of help when the more detailed techniques of gene cloning analysis are described. One of the striking aspects of gene manipulation technology is that many of the procedures can be carried out with a fairly basic laboratory setup. Although applications such as large-scale DNA sequencing and production-scale biotechnology require major facilities and investment, it is still possible to do high-quality work within a normal research laboratory. So the requirements can be summarized under three headings. First, general laboratory facilities. Second, cell culture and containment. Third, processing and analyzing. The overall containment requirements will usually be specified by national bodies that regulate gene manipulation, and these may apply to bacterial and mammalian cell culture facilities. Thus, a simple cloning experiment with E. coli may require only normal microbiological procedures, whereas an experiment to clone human genes using viral vectors in mammalian cell lines may require the use of more severe safety systems. So, cell culture and containment facilities are essential for growing the cell lines and organisms required for the research, with the precise requirements depending on the type of work being carried out. Most labs will require facilities for growing bacterial cells, with the need for equipment such as autoclaves, incubators, centrifuges, and protective cabinets in which manipulation can be performed. Mammalian cell culture requires a slightly more sophisticated facilities, and plant or algal culture usually requires integration of lighting into the culture cabinets. In many cases, some form of physical containment is required to prevent the escape of organisms during manipulation. The overall level of containment required depends on the type of host and vector being used, with the combination providing a level of biological containment in that the host is usually disabled and does not survive beyond the laboratory. Isolation of DNA and RNA. Every gene manipulation experiment requires a source of nucleic acid in the form of either DNA or RNA. It is therefore important that reliable methods are available for isolating these components from cells. There are three basic requirements. 1. Opening the cells in the sample to expose the nucleic acids for further processing. 2. Separation of the nucleic acids from other cell components. And 3. Recovery the nucleic acid in purified form. So a variety of techniques may be used, ranging from simple procedures with few steps up to more complex purification, involving several different stages. These days, most molecular biology supply companies sell kits that enable purification of nucleic acid from a range of sources. The first step in any isolation protocol is disruption of the starting material, which may be viral, bacterial, plant, or animal. The methods used to open cells should be as gentle and as possible, preferably utilizing enzymatic degradation of cell wall material and detergent lysis of cell membranes. If more rigorous methods of cell disruption are required, there is the danger of sharing large DNA molecules, and this can hamper the production of representative recombinant molecules during subsequent processing. Following cell disruption, most methods involve a deproteinization stage. This can be achieved by one or more extractions using phenyl or phenyl chloroform mixtures. On the formation of an emulsion and subsequent centrifugation to separate the phases, protein molecules partition into the phenyl phase and accumulate at the interface. The nucleic acids remain mostly in the operaceous phase and may be precipitated from solution using isopropanol or ethanol. Some techniques don't require the use of phenolic mixtures and are safer and more pleasant to use than phenol-based extraction media.
If a DNA preparation is required, the enzyme ribonuclease RNAs can be used to digest the RNA in the preparation. If mRNA is needed for cDNA synthesis, a further purification can be performed by affinity chromatography using oligo-DT cellulose to bind the poly-A tails of eukaryotic mRNAs. This gives a substantial enrichment for mRNA and enables most of the contaminating DNA, rRNA, and tRNA to be removed. As you can see here in figure 3.1, part A, total RNA is solution solution is passed through the column in a high salt buffer, and the oligo-DT tracks bind the poly-A tails of the mRNA. In part B, residual RNA is washed away with a high salt buffer, and in part C, the mRNA is eluted by washing with a low salt buffer. In part D, the mRNA is then precipitated under ethanol and collected by centrifugation. The technique of gradient centrifugation is often used to prepare DNA, particularly plasmid DNA or pDNA. In this technique, a cesium chloride solution containing the DNA preparation is spun at high speed in an ultracentrifuge. Over a long period, up to 48 hours in some cases, a density gradient is formed and the pDNA forms a band at one position in the centrifuge tube. The band may be taken off and the cesium chloride removed by dialysis to give a pure preparation of pDNA. As an alternative to gradient centrifugation, size exclusion chromatography or gel filtration or similar techniques may be used.
It is often necessary to use very small amounts of nucleic acid, typically micro, nano, or picograms, during a cloning experiment. It is obviously impossible to handle these amounts directly, so most of the measurements that are done involve the use of aqueous solutions of DNA and RNA. The concentration of a solution of nucleic acid can be determined by measuring the absorbance at 260 nanometers using a spectrophotometer, and a 260L1 is equivalent to a concentration of 50 microgram per ml for double-stranded DNA or 40 microgram per ml for single-stranded DNA or RNA. If the A of 280 is also determined, the A260 on A280 ratio indicates if there are contaminants present, such as residual phenol or protein. The A260 on A280 ratio should be around 1.8 for pure DNA and 2 for pure RNA preparation. In addition to spectrophotometric methods, the concentration of DNA may be estimated by monitoring the fluorescence of bound ethidium bromide. This type binds between the DNA bases and fluorescence orange when illuminated with ultraviolet or UV light. By comparing the fluorescence of the sample with that of a series of standards, an estimate of the concentration may be obtained. This method can detect as little as 1 to 5 nanogram of DNA and may be used when UV absorbing contaminants make spectrophotometric measurements impossible. Having determined the concentration of a solution of nucleic acid, any amount in theory may be dispensed by taking the appropriate volume of solution. In this way, nanogram or picogram amounts may be dispensed with reasonable accuracy. Precipitation of nucleic acid is an essential technique that is used in a variety of applications. The two most commonly used precipitants are isopropanol and ethanol, ethanol being the preferred choice for most applications. When added to a DNA solution in a ratio by volume of 2:1 in the presence of 0.2 molar salt, ethanol causes the nucleic acids to come out of solution. Although it used to be thought that low temperatures -20 or -70 were necessary, this is not an absolute requirement, and 0 centigrade appears to be adequate. After precipitation, the nucleic acid can be recovered by centrifugation, which causes a plate of nucleic acid material to form at the bottom of the tube. The plate can be dried and the nucleic acid were suspended in the buffer appropriate to the next stage of the experiment.
A major problem encountered in many cloning procedures is that of keeping track of the small amounts of nucleic acid involved. This problem is magnified at each stage of the process because losses mean that the amount of material usually diminishes after each step. One way of tracing the material is
to label the nucleic acid with a marker of some sort so that the material can be identified at each stage of the procedure. So, what can be used as the label? Types of label, radioactive or not. One way of tracing DNA and RNA samples is to label the nucleic acid with a radioactive molecule, usually a deoxynucleoside triphosphate or DNTP, labeled with tritium or phosphorus so that portions of each reaction may be counted in a scintillation counter to determine the amount of nucleic acid percent. This is usually done by calculation, taking into account the amount of radioactivity percent in the sample. Radioactive tracers have been used extensively in biochemistry and molecular biology for a long time, and procedures are now well established. The most common isotopes used are tritium, carbon, sulfur, and phosphorus. Tritium and carbon are low energy emitters, with sulfurous being a medium energy emitter and phosphorus being a high energy emitter. Thus, phosphorus is more hazardous than the other isotopes and requires particular care in use. There are also strict statutory requirements for the storage and disposal of radioactive waste materials, partly because of the inherent dangers of working with high energy isotopes. The use of alternative technologies such as fluorescent dyes or enzyme linked labels has become popular in recent years. Although these methods do offer advantages for particular applications, such as DNA sequencing, for routine tracing experiments, a radioactive label is still often the preferred choice. In this case, the term radiolabeling is often used to describe the technique. And labeling. In the labeling technique, the enzyme polynucleotide kinase is used to transfer the terminal phosphate group of ATP onto 5' hydroxyl termini of nucleic acid molecules. If the ATP donor is radioactively labeled, this produces a labeled nucleic acid of relatively low specific activity, as only the termini of each molecule become radioactive. As you see in figure 3.2, part A, DNA is dephosphorylated using phosphatase to generate 5' hydroxyl groups. In part B, the terminal phosphate of phosphorus ATP is then transferred to the 5' terminus by P and K. The reaction can also occur as an exchange reaction with 5' phosphate termini. Nike translation. Nike translation relies on the ability of the enzyme DNA polymerase I to translate a Nike created in the phosphodiester backbone of the DNA double helix. Nikes may occur naturally or may be caused by a low concentration of the nucleus DNase 1 in the reaction mixture. DNA polymerase I catalyzes a strand replacement reaction that incorporates new DNTPs into the DNA chain. and one of the DNA types supplied is radioactive, the result is a highly labeled DNA molecules. In figure 3.3, part A single-strand nick is introduced into the phosphodiester backbone of a DNA fragment using DNase 1. In part B, DNA polymerase I then synthesizes a copy of the template strand, degrading the non-template strand with its 5' to 3' exonuclease activity. If phosphorus DNTP is supplied, this will be incorporated into the newly synthesized strand. Labeling by primary extension. Labeling by primary extension refers to a technique that uses random oligonucleotides, usually hexadeoxyribonucleotide molecules, sequences of 6-deoxynucleotides, to prime synthesis of a DNA strand by DNA polymerase. The DNA to be labeled is denatured by heating, and the oligonucleotide primers annealed to the single-stranded DNA. The clonal fragment of DNA polymerase can then synthesize a copy of the template primed from the 3' hydroxyl group of the oligonucleotide. If a labeled DNTP is incorporated, DNA of very high DNA of very high specific activity is produced. In figure 3.4, part A, DNA is denatured to give a single-stranded molecule. An oligonucleotide primer is then added to give a short double-stranded regene with a free 3' hydroxyl group. In part C, the clonal fragment of DNA polymerase I can then synthesize a copy of the template strand from the primer, incorporating phosphorus DNTP to produce a labeled molecule with a very high specific activity. In a radiolabeling reaction, it is often desirable to separate the labeled DNA from the unincorporated nucleotides present in the reaction mixture. A simple way of doing this is to carry out a small-scale gel filtration step using a suitable medium. The whole process can be carried out in a Pasteur pipette, with the labeled DNA coming through the column first, followed by the free nucleotides. Fractions can be collected and monitored for radioactivity, and the data used to calculate total activity of the DNA, specific activity, and percentage incorporation of the isotopes. DNA sequencing. The ability to determine the sequence of bases in DNA is a central part of modern molecular biology and provides what might be considered the ultimate structural information. Rapid methods for sequence analysis were developed in the late of 1970, and the technique is now used in laboratories worldwide. In recent years, the basic techniques have been revolutionized by automation, which has improved the efficiency of sequencing to the point where genome sequencing is possible. According to Figure 3.5, individual bands or lane 1 indicate discrete fragments of DNA in this case. The fragments are
of phage lambda DNA caught with the restriction enzyme HND3. The sizes of the fragments in kilobases are indicated. The remaining lanes contain samples of DNA from an alga cut with various restriction enzymes. Because of the heterogeneous nature of these samples, the fragments merge into one another and show up as a smear on the gel. Samples that have migrated farthest, lane 3, 4, 5, 9, and 10, are made up of smaller fragments than those that have remained near the top of the gel for lanes 2, 7, 8, and 11. Principles of DNA sequencing. By definition, the determination of a DNA sequence requires that the bases are identified in a sequential technique that enables the processive identification of each base in turn. There are three main requirements for these to be achieved. First, DNA fragments need to be prepared in a form suitable for sequencing. Second, the technique used must achieve the aim of presenting each base in turn in a form suitable for identification. Third, the detection method must permit rapid and accurate identification of the bases. Principles of DNA sequencing. The sequencing protocol is essentially a technical procedure rather than the experimental one. There are several variants of the basic procedure, but the most widely used techniques are based on the enzymatic method. Whatever the method, the desired result is to generate a set of overlapping fragments that different terminate at different bases and different lengths by one nucleotide. This is known as a set of nested fragments. Assuming that the technique has generated a set of nested fragments, the detection step is the final stage of the sequencing procedure. This usually involves separation of the fragments on a polyacrylamide gel in which fragments are radioactively labeled. Generate an autoradiograph. Automated sequencing procedures tend to use fluorescent labels and a continuous electrophoresis to separate the fragments, which are identified as they pass a detector. There are two main methods for sequencing DNA. In one method, developed by Alan Maxson and Walter Gilbert, chemicals are used to clip the DNA at certain positions, generating a set of fragments that differ by one nucleotide. The same result is achieved in a different way in the second method, developed by Fred Sanger and Alan Coulson, which involves enzymatic synthesis of DNA strands that terminate in a modified nucleotide. Analysis of fragments is similar for both methods and involves gel electrophoresis and autoradiography, assuming that a radioactive a radioactive label has been used. The enzymatic method and variants of the basic technique has now almost completely replaced the chemical method as the technique of choice. Although there are some situations where chemical sequencing can provide useful data to confirm information generated by the enzymatic method. Preparation of DNA fragments. The difficulty of preparing a fragment for sequencing is largely dependent on the scale of the sequencing project. If the aim is to sequence part of a gene that has already been isolated and identified, the process is relatively straightforward and usually requires that the fragment is of a suitable length and in a suitable form for the sequencing procedure in use. If, however, the aim is to sequence a much larger piece of DNA, such as an entire chromosome, the problem is much greater. In this case, the sequencing strategy is important, and there are two approaches to this. The first is an ordered sequencing strategy, often called the clone by clone or clone contig method. Contig is an abbreviation of contiguous. The fragments are tracked as part of the shotgun approaches. A strategy and their relative order is noted as the project progresses. The sequence is put together by reference to the order of the fragments. The second approach is the so-called shotgun sequencing method, where the fragments are generated and processed at random. Assembly of the sequence is then carried out by searching for sequence overlaps using a computer. In Figure 3.6, Part A, an ordered approach is shown, where the relative positions of the clone fragments is known. This approach uses data from physical maps to enable clones to be assigned to particular parts of the chromosome. By extending this approach as cloned fragments become smaller and ultimately reach sequencing size, the sequence data can be assembled as a set of ordered fragment sequences. This approach is sometimes called the clone counting method. In Part B, the shotgun approach is shown. Here there is no attempt to determine the relative positional order of the sequence fragments. Instead, the fragments are generated and sequenced randomly, with the resulting sequence being assembled by matching origins of sequence overlap. The two approaches have their own merits. The order approach is more difficult at the stage of cloning and sequencing, but assembling the sequence is a little simpler than in the shotgun method. Maxam-Gilbert sequencing or chemical sequencing. A defined fragment of DNA is required as the starting material. This need not be cloned in a plasmid vector, so the technique is applicable to any DNA fragment. The DNA is radiolabeled with phosphorus at the 5' ends of each strand, and the strands are denatured, separated, and purified to give a population of labeled strands for the sequencing reactions. The next step is a chemical modification of the bases in the DNA strand. This is done in a series of 4 or 5 reactions with different specificities, and the reaction conditions are chosen so that, on average, Only one modification will be introduced into each copy of DNA molecule. The modified bases are then removed from their sugar groups and the strands cleaved at these positions using the chemical piperidine. The theory is that given the large number of molecules and the different reactions, this process will produce a set of nested fragments. In Figure 3.7, Part A, radio-labeled single-stranded DNA is produced. Part B, the bases in DNA are chemically modified and removed, with an average one base being affected per molecule. Part C, the phosphodiester backbone is then cleaved using piperidine. Part D, the process produces a set of fragments differing in length by one nucleotide labeled at their 5' termini. Sanger-Coulson sequencing. Although the end result is similar to that attained by the chemical method, the Sanger-Coulson procedure is totally different from that of Maxim and Gilbert. In this case, a copy of the DNA to be sequenced is made by the clonal fragment of the DNA polymerase. The template for this reaction is single-stranded DNA, and a primer must be used to provide the three terminus for DNA polymerase to begin synthesizing the copy. According to Figure 3.8, Part A primer is annealed to a single-stranded template, and the clonal fragment of DNA polymerase I is used to synthesize a copy of the DNA. A radio-labeled DNTP, often, often sulfur DNTP, is incorporated into the DNA. Part C, chain termination occurs when a dideoxynucleoside 3-phosphate or DDNTP is incorporated. Part D, a series of four reactions, each containing one DDNTPase in addition to the four DNTPase required for chain elongation, generates a set of radio-labeled nested fragments. The production of nested fragments is achieved by the incorporation of a modified DNTP in each reaction. These DNTPs lack a hydroxyl group at the 3' position of the deoxyribose, which is necessary for chain elongation to process. Such modified DNTPs are known as dideoxynucleoside 3-phosphates, or DDNTPs. The four DDNTPs A, G, T, and C are included in a series of four reactions, each of which contains the four normal DNTPs. The concentration of the dideoxy form is such that it will be incorporated into the growing DNA chain infrequently. Each reaction therefore produces a series of fragments terminating at the specific nucleotide, and the four reactions together provide a set of nested fragments. The DNA chain is labeled by including a radioactive DNTP in the reaction mixture. This is usually sulfur DATP, which enables more sequence to be read from a single gel than the phosphorus labeled DNTPs that were used previously. Concept map. The concept map of this class shows us that nucleic acids, either DNA or RNA molecules, may be arranged as complementary strands exploited in nucleic acid hybridization. These molecules also can be handled in solution but may be precipitated using ethanol or isopropanol. DNA or RNA molecules can be separated by electrophoresis on the basis of fragment length. DNA can be sequenced by two methods: chemical method or Maxam-Gilbert, and enzymatic method or Sanger-Coulson, which generate nested fragments and will be separated according to the fragment length. DNA or RNA molecules can be radiolabeled by end labeling or strand labeling by two methods: night translation and primer extension. Thank you for attention and see you next time.